All right, your Unit 3 exam on genetics and biotechnology is right around the corner. So let's make sure you walk in there feeling like you've totally got this. We're going to nail down the key concepts you absolutely need to know.
And that's exactly what we're about to do. We're going to cut through all the extra noise and just focus on the essentials. Think of this as your final high-speed review to really lock in everything you'll need.
So, first things first. We have to start at the very beginning, with the master blueprint for pretty much every living thing, DNA. This is where the story of you and every other organism really begins.
We've all seen this shape, the iconic double helix. But the real magic, it's on the inside. It's the specific order, the sequence of those 4 chemical bases, A, T, C, and G, that works like an instruction manual. This tiny little code dictates literally everything about you, from your eye color to how your cells do their jobs.
Okay, pay attention here because this is one of the most important things you need to know for your exam, the central dogma. Just think of it like this. DNA holds the master plan. RNA is like a messenger that carries a temporary copy of that plan. And proteins? They're the machines that actually go out and do the work. This flow, DNA to RNA to protein, is so fundamental. It's how a cell keeps its original blueprint safe and sound in the nucleus while sending out disposable copies to the factory floor.
Now this brings up a classic exam question, one that almost always shows up in some form. You've got to be able to clearly explain the difference between the master blueprint, DNA, and its messenger, RNA. They're partners for sure, but they are not the same thing.
And here they are, the 3 key differences you need to memorize. First, the sugar. DNA has deoxyribose, RNA has ribose. Easy. Second, the structure. DNA is that famous double helix, 2 strands, while RNA is usually just a single strand. And third, the bases. They both use A, C, and G, but DNA uses thymine, T, while RNA swaps that out for uracil, U. Seriously, burn this chart into your brain. It is pure gold for your test.
Okay, so we've got the code, now what? The next critical piece of the puzzle is how the cell actually reads that code and turns it into something useful, like a protein.
This is really the million-dollar question, isn't it? How does a sequence of letters locked away in a cell's nucleus get turned into the actual machinery that keeps you alive? Well, it happens in 2 main steps. And here they are, transcription and translation. First, you transcribe the message, then you translate it. A good way to remember this is to think about copying a recipe from a fancy cookbook, that's transcription, and then taking that copy into the kitchen to actually bake the cake, that's translation.
Let's dive into step 1, transcription. This whole process happens inside the cell's nucleus. You can see here how this enzyme, RNA polymerase, comes along, unzips a small section of the DNA, and then builds a copy using RNA. That new copy is called messenger RNA, or mRNA for short. It's our message.
So now that we have our mRNA copy, its whole job is to be the messenger. It's got to get that precious genetic information out of the protected nucleus and into the main part of the cell, the cytoplasm. It's looking for the ribosomes, which are basically the cell's protein-making factories.
So let's really nail this down, because comparing these 2 processes is another guaranteed exam topic. Transcription, copying DNA into RNA. It happens in the nucleus. Translation, reading that RNA to build a protein. It happens at the ribosome, out in the cytoplasm. Location and function, know the difference.
Now, a cell has thousands and thousands of genes, right? But it doesn't need them all turned on at the same time. That would be chaos. This brings us to a super important idea, gene regulation. It's all about how a cell decides which genes to turn on and which ones to keep off.
And honestly, the best way to think about this is like a simple light switch. Your muscle cells and your nerve cells have the exact same DNA blueprint. The reason they look and act so differently is because they flip different switches. They turn on different sets of genes. This control is absolutely everything.
So how does the cell actually flip these switches? It uses a few key tools. Promoters are like the start here sign right before a gene. Transcription factors are the proteins that act like a hand on that light switch, either helping to turn a gene on or blocking it to keep it off. And in bacteria, you have these cool things called operons, which are like a power strip that can turn a whole group of related genes on or off all at once.
So far, we've been talking about this system like it runs perfectly. But what happens when there are mistakes? When the genetic code gets changed, well, we call that a mutation.
A key thing to get straight is that a mutation is just a change in the DNA sequence. And it's really important to remember that not all mutations are bad. Some don't do anything at all. Some can be harmful, for sure, but every once in a while, a mutation might even be beneficial.
Let's compare two major types, another common exam topic. In a substitution, one letter just gets swapped for another. This might change a single amino acid in the protein, or it might do nothing. But a deletion, where a letter is completely removed, that's often way more serious. It can cause something called a frame shift, which messes up how every single letter is read from that point on. It basically turns the rest of the message into gibberish. And that's why deletions can be so much more damaging.
All right, let's switch gears. We've talked about how the code works naturally. Now let's talk about how we humans have learned to read it, copy it, and even edit it. Welcome to the world of biotechnology.
One of the most important tools in the box is PCR. You can think of it as a molecular copy machine. Let's say you have just a teeny tiny speck of DNA from a crime scene or an ancient fossil. PCR lets you amplify that, making millions, even billions of copies so you actually have enough to study.
So what do you do with all those copies? Well, one incredible application is DNA fingerprinting. This image here shows the end result. It's a process that separates DNA fragments by size, and it creates a unique pattern of bands for every single person. It's like a genetic barcode. You can see how forensics can compare the pattern from crime scene evidence to a suspect's pattern to find a match.
And it's not just about solving crimes. Biotechnology is completely changing medicine. We can now test for genetic diseases before they show symptoms, we can understand a person's risk for certain conditions, and we can even start developing therapies that are custom-made for an individual's unique genetic code.
But of course, with all this incredible power come some very serious questions. As we get better and better at reading and rewriting our own DNA, we run into some really complex ethical, legal, and social issues. Who should have access to your genetic information? How do we stop that information from being used for discrimination? And where, exactly, do we draw the line?
And that's really the big question to leave you with. We are standing on the edge of a new frontier with the power to literally rewrite the code of life itself. And we have to ask ourselves, just because we can do something, does that mean we should? And who gets to make the rules? It's something to think about long after you ace that exam. Good luck.